Hey, this is Mrs. Dellosso, and in this video we're going to talk about the cell cycle. So let's talk about the goals that you need to understand and know for this video. So first thing we need to talk about and know is the cell cycle, and there are three main stages of the cell cycle that we're going to get into. First one is interphase, second stage I would talk about is mitosis, and third, cytokinesis. Second thing we're going to talk about are the uses of mitosis, and finally the last thing that we'll talk about are the types of asexual reproduction. So make sure you get those goals down and then we'll get started. Alright, so let's start talking about the cell cycle. The purpose of the cell cycle is we're going to talk about how cells go from 1 to 2 cells and continue reproducing from that point. So the cell cycle is really going to talk about prepping for the actual cell division, and we're going to talk about what else needs to take place in order for the cell to actually divide. And if you take a look at the top here we're showing or looking at a cell, one single cell undergoing the cell cycle or cell division here. So one cell is going to divide into two, then these cells, after the first cell division, each of these new cells can divide into two, which will make 4 new cells, and so forth. So this is the process that we're going to be talking about. First stage that we mentioned before is interphase. Interphase is going to be the time of growth for the cell, prepping for mitosis. Mitosis is simply the division of the nuclear material. What is nuclear material? It's also known as DNA, genetic information. Following mitosis will be cytokinesis, and that is the final stage which will actually separate the two new cells. So that's going to be division of the cytoplasm. So let's take a look at what interphase is all about. Alright, so let's talk about interphase. Again, interphase is basically a prep, getting ready for mitosis. So let's just refresh our memory on some of the structures here. Here's our cell. Right over here is going to be the nuclear membrane that surrounds the genetic material. This loosely wound DNA, this is going to be referred as the chromatin, not yet chromosomes. And over here we'll have our centrioles, which will play a key part in cell division. And keep in mind the function of the centrioles as well is not really known altogether just yet, but they do know that it has to do with cell division. Alright, so let's go a little bit more in depth with interphase. Alright, so interphase. Let's take a look at this chart over here to your right. This chart here represents the whole entire cell cycle. And if you notice, we see interphase here, here, and over here. So interphase takes a lot of the cell cycle, meaning the cell spends most of its time in interphase. And if you notice here, interphase is actually divided up into 3 other phases, which we're going to talk about right now. So the first phase of interphase is the G1 phase, and this is just a growth phase. This is when the cell is going to grow, increase in size, prepping for cell division. The second phase is the S phase, and this is very important because this is when DNA replicates. Why is DNA replication important? It's important because that's going to ensure that the 2 new cells that form will have an exact copy of DNA. Without any DNA, the cell's not going to have any instructions or know what to do. Also, during this phase, the centrosomes will divide, and the centrioles, those structures that we just looked at in the previous slide, will appear. Alright, third phase of interphase is the G2 phase, and this is the last phase in for preparation for mitosis. Alright, so that covers interphase, the first stage of the cell cycle. Alright, so next up, the second stage of the cell cycle that we're going to look at is mitosis. Now, if you take a look at this chart here, this green area, that all represents interphase. But now we're going to take a look at this chunk over here, where it says mitosis. Now, just like interphase had 3 different phases, mitosis has its own phases as well. Mitosis has prophase, metaphase, anaphase, and then telophase. Cytokinesis will be the very, very last step in this process. So let's take a look at these 4 different phases of mitosis. Alright, so let's take a look at the first phase of mitosis, and that's prophase. So if you notice over here in prophase, we have these spindle fibers that are going to start to form. Our centrosomes with the centrioles will move to the ends of the to either side of the cell here, if you notice, the nuclear membrane starts to break down. Or disintegrates. And also, we no longer see chromatin, but now we have these nice tightly condensed chromosomes. And because each of these chromosomes are attached here, remember, DNA is already duplicated, so they're attached to their sister chromatids. So over here we have one chromosome, and attached to it, it's its identical copy, and that's the chromatids, the sister chromatids, and those are attached by the centromere. So the main thing about prophase is that the nuclear membrane starts to break down, and now the chromatin has condensed into chromosomes, and the sister chromatids are matched up. All right, next in mitosis is called metaphase, and we call it metaphase because we think meta, middle. Now the sister chromatids, if you take a look, are lined up in the middle here along the equatorial plate. And the spindle fibers, some of them are actually attached to the centromeres of the sister chromatids, and I want you to take a look. Think about why that might be important as far as cell division. Some of the spindle fibers, they aren't attached. They're just going to help with keeping the cell elongated or large, and the centrosomes are located over here. So main thing for metaphase is that the sister chromatids are lined up right in the middle of the cell. All right, next we have anaphase, and during anaphase, the sister chromatids are actually going to separate, and they're starting to move along these spindle fibers. Now, some scientists have found that it's almost like, think of this chromosome right here as Pac-Man. There's some proteins that might be going down or like consuming those spindle fibers to move down to separate there. So you can think of it that way. And this ensures that each of the new cells that will get produced will have an exact same amount of chromosomes. So that's why the sister chromatids are going to start to separate during anaphase. Right after anaphase, we have telophase, and already we can start to see the cells start to separate. Right now we're taking a look at an example of an animal cell here, and this pinching that starts to form here, that's called the cleavage furrow. And that's where the cell is actually going to divide. This is also where we start to see the nuclear membrane reappear. The cells aren't completely separated just yet, but each of these nuclei contain the same exact genetic information. And finally, after mitosis is complete, we'll start to see cytokinesis. And cytokinesis is the division of the cytoplasm. And as you can see, as a result of cytokinesis, we have two new genetically identical daughter cells. So these two cells formed from one single cell. So in other words, one single cell divided into two new cells, and because these cells came from one cell, they're genetically identical. So that means they have the same exact DNA in both of these cells. So cytokinesis is the end result of the cell cycle. It's division of the cytoplasm, and from that we have two new cells. All right, so let's get into the uses of mitosis here. If you take a look at this diagram, we're starting with a zygote, which is a fertilized egg cell or one cell. And through the process of mitosis, we're producing more and more cells. And this organism is a sea star, which is made up of many, many cells. So in order to go from one cell to many cells, to have growth, our cells go through the process of mitosis. The same thing happened when we were conceived. We started out as a single cell after the sperm and egg joined together, and through the process of mitosis, you began to grow and develop more cells. So one of the major uses for mitosis is growth. The second thing is actually for repair or to replace worn out cells. In other words, certain cells can only last a few days, some weeks, maybe some even months. But after those cells have reached their full lifespan, there has to be more cells that are made to continue to do the job to help keep you alive. So for example, some red blood cells, in order to produce more cells, they'll undergo the process of mitosis. So growth is one use for mitosis. Repair, or to replace worn out cells, is another use for mitosis. And it's also a way of reproduction for single-celled organisms such as this amoeba here. Single-celled organisms can use the process of mitosis in order to produce more of their kind. So this amoeba will undergo mitosis. So this one-celled structure, now divided, now is into two new amoebas. Alright, so let's talk about types of asexual reproduction. So mitosis is one form of asexual reproduction, starting with one cell and producing two new cells that are genetically identical. So anything that reproduces asexually is going to be genetically identical to the parent, meaning it's going to have the same exact copy of DNA. So that means that we weren't produced asexually because we're not identical to either one of our parents. We're actually a combination of the two. So one example that we've been looking at of asexual reproduction is mitosis. A second type of asexual reproduction is something called binary fission. And that's the same thing that we looked at with the amoeba. That's how single-celled organisms can reproduce, starting with one cell and ending up with two new daughter cells here. That's stage 4. So here are two new cells. So binary fission, starting with one parent cell, such as the amoeba, and then ending up with two new daughter cells. Other types of asexual reproduction are spores, organisms that produce by making spores, such as like molds. That's how they can reproduce. Those spores will be released. Those are produced through mitosis. And this last type of asexual reproduction that we're going to talk about is budding. This is when the daughter cell kind of buds off from the parent cell, and even though it may look smaller than the parent cell, it's still genetically identical, even though it's smaller. And some example organisms that can do this are Hydra and yeast. So they can also do that by budding. And the daughter cells can remain attached, or sometimes they'll become unattached, but they're still genetically identical to the parent. Okay, so that sums up the cell cycle and mitosis, and also the uses of mitosis. So make sure you get all those notes down. If any questions, just bring them with you to class.